Today, we'll conclude our 2 lectures on biological models and experimental techniques. By the end of this lecture, you should be able to identify equipment used in anaerobic culture. You should be able to explain the challenges of making anaerobic media, and you should be able to outline the steps of transferring a broth culture of bacteria to a new tube. Your reading is Chapter 7 from the textbook. From an earlier lecture, you recall the reasons why oxygen is toxic to anaerobes. Superoxide and hydrogen peroxide are toxic forms of oxygen in the cell. Superoxide dismutase and catalase serve as defenses against these toxic forms, but many anaerobes lack these defensive enzymes. You also recall that oxygen is toxic to anaerobes, but even so, low oxygen is not enough for anaerobes to grow. The environment is still too oxidizing, even in the virtual absence of oxygen. The redox potential of the rumen being far lower than media equilibrated with a nearly oxygen-free atmosphere, containing 1 molecule of oxygen per liter. Because of the sensitivity of anaerobes to oxygen and high redox, the goal of anaerobic culture is to maintain oxygen-free and low redox conditions. We have to maintain these during handling of cultures and media preparation. This goal is conceptually quite simple, but the equipment we have to use to accomplish this goal can be rather complex. It might remind you of a Rube Goldberg machine, as we see on the right. Rube Goldberg was a cartoonist who made a career out of illustrating very complicated machines that accomplished very simple tasks. The example machine on the right shows a self-operating napkin, and if you follow all the steps labeled A through M, you can see how it works. When we discuss equipment used for anaerobic culture, try to keep the larger goal in mind and don't get lost in the complicated steps. The gassing station is the centerpiece of any anaerobic lab. It dispenses oxygen-free gas through gassing probes and into culture tubes or bottles. The gassing station we see on the left is connected to CO2, nitrogen, or hydrogen tanks. The flow of that gas is controlled at the gassing station using valves. The gas is eventually routed to a gassing probe as we see at the right. This gassing probe fits into the neck of culture tubes and bottles and allows them to be continuously flushed with the oxygen-free gas. The gas is eventually routed to a gassing probe, which fits into the neck of culture tubes or bottles and permits them to be flushed with oxygen-free gas whenever they're opened. This prohibits the entry of oxygen. The bottle illustrated at right is currently empty, but would be filled with culture or media during routine use. Culture bottles, like that on the previous slide, and culture tubes, like that on the current slide, are constructed to minimize the entry of oxygen when they're closed. They're constructed with thick glass, which is impermeable to oxygen, and they're stoppered with butyl rubber. Butyl, not standard rubber, is used because the former is less permeable to oxygen. These tubes and bottles can be used either with a broth or an agar culture. For agar tubes, the agar is coated on the walls to provide a surface for microbial growth. Working with tubes that have agar coated on the walls is not particularly easy. You can see at right how we pick colonies from the tubes using a Pasteur pipette. It takes a steady hand to pick the colony that you want without contaminating the entire tube. Fortunately, there's an alternative to using these tubes for agar culture, the bottle plate. The use of these bottle plates is analogous to petri dishes used in aerobic culture. The figure at the right shows that the bottle plate provides a broad, flat surface for agar, similar to the petri dish. The bottle is normally stoppered, but during use, one of the stoppers can be removed, and the bottle can be flushed through the other stopper with gas from the gassing station. Colonies that grow on the agar can be picked using a long needle. Syringes and needles are important to anaerobic culture. They're used to penetrate the stopper of a culture tube or bottle during the transfer of a microbial culture to a new tube. Their use serves as an alternative to opening and closing tubes under the gassing probe, which can lead to contamination. The figure at the right shows how a syringe and needle are used, and we'll see more in the demonstration video of how to transfer a bacterial culture. We'll return to the gassing station because there's one detail that we didn't cover earlier. That's the oxygen scrubbing column that we see in the red box. We use an oxygen scrubbing column because high-purity commercial gas still has some oxygen. The grade we use has 20 parts per million. This may not seem like much, but any oxygen is not tolerable in anaerobic culture. The oxygen scrubbing column contains copper and it's heated. The CO2 is passed through this heated copper to remove the last traces of oxygen. You can see in the equation that copper from the column combines with oxygen to form copper oxide. With the oxygen now removed, the CO2 is ready for anaerobic culture. After a time, all the copper in the column will react and form copper oxide, and occasionally the column has to be regenerated by passing hydrogen through it. In the equation, we can see that copper oxide reacts with hydrogen to reform copper. It also produces water. An alternative to the gassing station is a glove chamber. This permits petri dishes and traditional methods of culture to be used with minimal modification. All materials are introduced into the chamber using an airlock, and the ports fitted with gloves allow materials to be manipulated inside the chamber. Instead of an oxygen scrubbing column, the chamber uses a palladium catalyst to remove trace amounts of oxygen. Oxygen combines with hydrogen, which is present in the mixed gas in the chamber, to form water, keeping the concentration of oxygen low inside the chamber. A glove chamber may seem like a great alternative to a gassing station, but in practice it's very difficult to keep leak-free. In the figure at the right, the chamber closest to you has a severe leak. The chamber cannot even be kept inflated, and you can see that the top is tenting downwards. These leaks are nearly impossible to find and to fix. The lab that owns this chamber tried several times, and it couldn't make the repair. It continued to use this chamber, but only for non-critical work. For aerobic microbiology, preparing media is a trivial exercise. You combine components together, bring them up to volume with water, and autoclave to sterilize. If that seems like too much work, you can even buy media pre-made. This is 1x LB broth. Simply pour and use. In anaerobic microbiology, we're not so fortunate. Preparing media is not a trivial exercise. It takes hours or days to make a single batch, and we have few commercial sources for buying pre-made media. The first step of media preparation is to boil. Gas is theoretically insoluble at 100 degrees Celsius, which should drive off all the oxygen. In practice, not all the oxygen is boiled off, and we have to bubble with CO2 for hours to drive off even more oxygen. As we discussed before, removing oxygen is not enough. The redox has to be lowered to a level that's tolerable for anaerobes to grow. So next, we reduce the media, we lower the redox with a reducing agent, such as cysteine, sulfide, or hydrogen. We see the addition of the reducing agent with a syringe at the bottom. You may have noticed in the last slide that the media was blue. Now it's colorless. Earlier, we added a redox indicator, rizazurin, which is blue when it's oxidized. It turns pink and then colorless as it's reduced. The fact that the media has turned from blue to now colorless means that it's fully reduced. Removing the oxygen was not enough, we had to add the reducing agent. Now that the media is reduced, we can finally dispense and autoclave to sterilize it. But even this step is non-trivial. We have to keep the bottles and tubes stoppered during autoclaving, or we undo all the work that we did. As you know, you're not really supposed to autoclave a closed bottle. For safety, we put the bottle in a metal shield. That way, if it explodes, the shield will absorb the shrapnel. In the last part of the lecture, we'll watch a video demonstration for how to transfer a bacterial culture from an old tube to a new tube of media. This is a routine part of anaerobic microbiology. It must be done when you want to start an experiment, And also, it must be done to simply maintain a culture. If a culture is not transferred every couple of weeks, it will lose viability. To transfer an anaerobic culture, we first turn on the CO2 tank. Next, we turn on valves on the gassing station to start the flow of CO2. After that, we turn on the Bunsen burner, just as we do in aerobic microbiology. We retrieve a new gassing probe that has been autoclaved in aluminum foil in order to sterilize it. We remove the old gassing probe, which is no longer sterile. We exchange it with the new, sterile one. We flame the needle of the probe with a Bunsen burner. It should still be sterile from autoclaving, but we flame it as a precautionary measure because it's easily contaminated. We retrieve 2 tubes, one containing the old culture and one containing fresh media. We retrieve a 1 milliliter syringe and needle, which we will use later to transfer the culture. We sterilize the rubber stoppers of the tube by spraying them with 70% ethanol and igniting the ethanol with a Bunsen burner. We do this for both tubes. Now we're ready for our syringe. We attach a needle to the syringe aseptically. In order to flush out trace amounts of residual air, we must flush out the syringe with CO2 by inserting into the gassing probe and pulling the plunger in and out. We pierce the tube with the old culture, which has a pink label. We withdraw a small volume, about 0.1 milliliters. We take the tube of new media and inject the culture using the syringe. This completes the transfer. We will turn off the gassing station, incubate the tube in a 39-degree incubator overnight, or until the culture grows. We will repeat this transfer when we want to start an experiment, or after a few weeks in order to maintain viability of the culture. Today we inoculate only 1 tube of media, but normally we inoculate at least 2 tubes in order to increase the volume and protect against tubes that don't grow. The take-home messages from this lecture are that anaerobic culture must maintain reducing conditions at all times. The gassing station, or glove chamber, is the centerpiece of an anaerobic lab and where all procedures are performed. Finally, transferring and handling cultures requires a series of coordinated steps.